Good morning, everybody. Thank you for joining us here for the online Immunology Journal Club. I'm Diana, and I'm going to be talking about culturing and gene editing of antibody-secreting B cells. So a quick disclaimer is that the publications that I'll be looking at today are both open access and they're licensed under the Creative Commons Agreement. So to give you some background on B cells are produced from hematopoietic stem cells in the human bone marrow. These cells then egress the bone marrow and travel through the blood into lymph nodes and other lymphoid organs. Here, naive B cells can follow different pathways and trajectories, one of which is to form memory B cells or antibody-secreting cells. Antibody-secreting cells are unique in their ability to produce large amounts of antibody, and they do this in the blood. These cells are fairly short-lived unless they travel into the bone marrow, where they can transition into being long-lived plasma B cells. So along the way, some functions of these B cells is cytokine secretion, antigen presentation to other immune cells, and importantly, long-lived plasma B cells and antibody-secreting cells are the main source of long-lived protective antiviral antibodies, where these cells can produce hundreds to thousands of antibodies per cell per second. So because plasma B cells can survive for decades and they can produce large amounts of proteins, which are not limited to just antibodies, this makes them very attractive very attractive for therapeutic applications. So some examples of these applications are involving gene editing of plasma cells, and these can be gene edited to express antigen-specific antibodies, which can be specific to an epitope on either tumors or viruses. In addition to this, B cells can be edited to express survival or engraftment factors, such as B cell activating factors, like BAFF, that can help these cells when they're actually engrafted into an individual. Other therapeutic factors that B cells can secrete can include, for example, factor VIII for patients with hemophilia B. So all of these applications make these cells quite attractive targets for therapeutic applications. So the first paper that I'll be talking about looks at studying the biology of antibody-secreting cells, and this paper is by Dawn Nguyen and her colleagues. It's published in Nature Communications in 2018, and the title of this paper is Factors of the Bone Marrow Microniche that Support Human Plasma Cell Survival and Immunoglobulin Secretion. So first, I'm going to take you through some background. So as we already know, in vivo, circulating antibody-secreting cells can become long-lived plasma cells, which reside in the bone marrow, and these are supported by niche cells. Long-lived plasma cells are capable of providing protective immunity for decades, and the ultimate goal of many vaccines is to generate long-lived plasma cells to confer long-term protection. Currently, it's unknown what niche factors are required to culture long-lived plasma cells in vitro. And furthermore, it's also difficult to culture antibody-secreting cells in vitro, making it even more difficult to study that transition of ASC to LLPC. Thus, it's important to identify niche factors that are required for this transition in order to elucidate the mechanisms and eventually harness the potential of these cells in therapeutics. So in this work, authors describe a novel culture system which they can use to test known as well as identify novel niche factors which are required for antibody-secreting cell survival. So first, I'm going to take you through the co-culture system that they use in the majority of their experiments. And in this culture system, they isolate primary bone marrow mesenchymal stromal cells from healthy subjects. And they isolate this by sorting these cells on CD90 positive, CD73 positive, CD45 negative, and CD19 negative markers. From there, these cells are either used directly in co-culture with other cells, or they're used to generate the secretome, which is essentially a conditioned media, which is collected off of these MSCs. The antibody secreting cells from the blood are also isolated from healthy subjects in either steady state or post-vaccination, which is used to boost the number of antibody secreting cells in the blood. They're then isolated from PBMCs by sorting on CD19 positive, CD38 high, and CD27 high cells. From there, the ASCs are either directly co-cultured with the MSCs, they're cultured in the secretome, which was generated from the MSCs, or they're cultured in the media only. And the media that they use here is RPMI with 10% heat inactivated FBS. These cultures are then assessed on different days in order to enumerate the number of live and antibody secreting ASCs by the ELISpot assay. So one of the first things that the authors did was to actually show that MSCs themselves in a co-culture are able to support antibody secreting cell survival in vitro. So what you see in A is that when antibody secreting cells are cultured just in media, they diminish by day 2. However, when they're co-cultured with mesenchymal stromal cells, they're able to survive. 50% of cells are able to survive up until about day 7. They then used irradiated MSCs in order to demonstrate that survival can be prolonged for longer than just 7 days. And they used MSCs from 2 different subjects, one in healthy state and one post-vaccination. And you could see that the trend is quite similar for both of the subjects, where by day 31, you get about 15 to 20% of ASC survival when co-cultured with mesenchymal stromal cells. Further, they were interested in elucidating whether cell-to-cell contact was important in this for the survival of ASCs. And they did an experiment where they cultured ASCs in media alone, in co-culture as done before, through a membrane and a transwell experiment, and with just the secretome, which was generated from MSCs. And they show here that cell-to-cell contact is not required for the first 6 days of culture. As you can see on days 1, 3, and 6, there's no significant difference between co-culture, transwell, or secretome culture. They then wanted to demonstrate that secretome was also able to support ASCs in long-term ex vivo culture, and they show this for up to 56 days. That the co-culture of the secretome supports about 20% of cells by day 30, and this is similar to what they see with the direct co-culture with MSCs. They noted in all of their previous experiments that they see a significant increase of IgG secreting antibody secreting cells in the first 72 hours. And the authors hypothesized that this would either be due to rapid proliferation in the first 3 days or the restoration of IgG secretion. And they hypothesized this because they suspected that sorting the cells would cause the antibody secretion function to be reduced. And so in A here, they show that at the sort time, when they look at their antibody secreting cell population, almost 95% of these cells are Ki67 positive, suggesting that most of these cells were recently rapidly proliferating. They also show that when you compare equal numbers of PBMCs, that sorting actually diminishes your percentage of IgG antibody secreting cell survival, demonstrating that the sort seems to cause a great reduction on the first day. Because they knew the cells were rapidly proliferating initially prior to the sort, they also wanted to see what was happening in vitro. So they performed a BRDU incorporation assay, and in the antibody secreting cells plus the secretome, BRDU was actually not being incorporated, suggesting that these cells are not actively dividing when being cultured in vitro. And they did a control to show that their IMSCs and secretome were not actually contaminated with any antibody secreting cells, and that any spots they were seeing in their IgG LE spot were actually due to their isolated antibody secreting cells. So from here, they were able to conclude that the early increase that they see is due to recovery of the secretory function and not actually proliferation. Further, they wanted to test APRIL, a proliferation-inducing ligand, which is a factor which is well established in the survival of long-lived plasma cells. They wanted to see if this factor would also have an impact on the survival of antibody-secreting cells. Initially, they noticed that adding APRIL to media alone had no impact on survival. It was still very low. However, when you supplemented the secretome with APRIL, you see that APRIL can enhance 7-day survival about twofold. They also tried supplementing media alone with other factors, such as B-cell activating factor BAFF, IL-5, IL-6, and IL-6 alone, and none of these provided any survival benefit on days 1, 2, and 3. When they wanted to look at the effect that APRIL has on the secretome long-term, they noted that although not as significant, the survival benefit of having APRIL in the secretome is actually sustained for up to 50 days. They also tried to add other single factors to the secretome, such as BAFF, IL-6, IL-5, interferon gamma, VEGF, or FGF, and these had no survival benefit. However, from previous publications, the authors suspected that IL-6 was important and was already contained in the secretome, and they tested this by adding an anti-IL-6 antibody to the secretome. And when they tested this at days 1, 3, and 7, they noted that the survival of antibody secreting cells was significantly reduced with the addition of the antibody, suggesting that IL-6 is an essential component of the secretome, but does not need to be further supplemented when the secretome is there. So now that they've demonstrated that their method is able to identify whether factors are critical or not for survival of antibody secreting cells, they wanted to identify some novel factors. And they wanted to do this by doing proteomic analysis on different secretome fractions. So they performed a few experiments to identify which secretome fractions were active versus non-active, active being the fractions that support antibody secreting cell survival, which are shown here in blue, and non-active, which don't support ASC survival, which are shown here in black. So in the first experiment, they show that secretomes from irradiated or non-irradiated MSCs can support antibody secreting cell survival as they've shown before. They show again that media alone does not support the survival of antibody secreting cells. And interestingly, when they ultracentrifuge the secretome to generate a supernatant, they show that supernatants of irradiated MSCs does support the survival. However, non-irradiated supernatant does not. They performed an additional experiment where they compared the MSC secretome to the blood secretome. The blood secretome was generated from blood adherent cells, which were cultured to generate a conditioned media. And they show here that blood secretome does not support antibody secreting cell survival. So they performed proteomics on all 5 fractions, so 3 active and 2 non-active fractions. And what they then did is they overlapped these in order to identify which components of the secretome are actually critical to sustain the survival of these cells. So here they overlapped this and they were able to identify 91 overlapping proteins. They then wanted to identify potential protein-protein interactions, and for this they used HIPI, which is a human integrated protein-protein interaction, or efferents. So this allowed them to identify over 4,000 potential protein-protein interactions with these 91 proteins. They wanted to further filter this, and in order to do this, they wanted to identify which of these were actually important for the transition of antibody-secreting cells to long-lived plasma cells. So they took advantage of already existing transcriptomics data from blood antibody-secreting cells and compared it to bone marrow long-lived plasma cells. And from here, they identified over 2,500 differentially expressed genes. When overlapping these differentially expressed genes with the potential protein interactions, they were able to come up with 556 overlapping gene-protein targets. These gene-protein targets were further filtered down by identifying genes or proteins with the most potential protein interactions. Then the whole list, or the filtered list, was analyzed using gene set enrichment analysis, which I'll get more into later. And the factors identified by these factors and pathways were then validated in their co-culture system. So when they performed, after the overlapping of genes and proteins, they had 12 proteins which passed the cutoff of having at least 45 potential protein interactions, or PPIs. And the top two, fibronectin and 14-3-3 zeta delta, or YWHAZ, had almost 120 different protein-protein interactions. This is not surprising, as both of these interact with a lot of different proteins. For example, YWHAZ is involved in signal transduction in many pathways, including cell survival. Further, their gene set enrichment analysis data of overlapping gene-protein targets identified 20 statistically significant hallmark pathways, and these pathways included mTOR complex 1 signaling, PI3 kinase, AKT mTOR signaling, and hypoxia. Out of the 20 identified pathways, fibronectin and YWHAZ were involved in 10, including the mTOR signaling, but not including the hypoxia. So after this, they wanted to validate their targets, and they were able to show that the two proteins that they identified in their proteomic transcriptomic overlap of fibronectin and YWHAZ, that these are essential components of the MSC secretome. And they showed this by adding anti-fibronectin and anti-YWHAZ antibodies, and showing that adding these antibodies significantly decreases antibody-secreting cell survival on days 1, 3, and 7. In addition, because the proteomics transcriptomics suggested that mTOR pathway downregulation was involved in the transition from antibody-secreting cell to long-lived plasma cell, they wanted to try a mTOR inhibitor on either antibody-secreting cells and long-lived plasma cells. And they noted that downregulation of mTOR had reduced the survival of antibody-secreting cells, but not long-lived plasma cells, which is consistent with the hypothesis that the mTOR pathway is downregulated through the transition from antibody-secreting cell to long-lived plasma cell. Further, they identified hypoxia, which was thought to be independent of fibronectin and YWHAZ, as they weren't involved in those pathways. And they tried culturing their secretome and antibody-secreting cells in hypoxic conditions. Hypoxia, when combined with the secretome alone, had a very small effect, where it increased the survival of antibody-secreting cells from 9 to 18% by day 14. However, when hypoxia was combined with APRIL, it actually had quite a significant enhancement of survival even in long term, where it was enhancing antibody-secreting cell survival to 40% at day 56. To conclude the key findings from this paper, bone marrow-derived mesenchymal stromal cell secretome is alone able to support about 20% of antibody-secreting cells in vitro for up to 30 days. They then used integrated bioinformatic analysis of mesenchymal stromal cell secretome proteomics and transcriptomics of antibody-secreting cells and long-lived plasma cells, which allowed them to identify novel proteins and pathways, which play key roles in bone marrow microenvironment. Fibronectin-1 and YWHAZ were then tested in their co-culture system and concluded to be critical for the survival of antibody-secreting cells. And further, hypoxia, which was identified as an important pathway, works synergistically with APRIL to actually significantly increase antibody-secreting cell survival in a long-term culture of 56 days. So from here, I'm going to move to the second paper, which deals more with editing antibody-secreting cells. And this paper is published by Harald Hartwager in Journal of Experimental Medicine in 2019. And it's titled HIV-Specific Humoral Immune Responses by CRISPR-Cas9 Edited B Cells. Some background for this paper: a small proportion of human immunodeficiency virus, HIV patients, develop broadly neutralizing antibodies, which occur naturally after 1 to 3 years after infection. These broadly neutralizing anti-HIV antibodies have been shown to suppress viremia in humans, and there are ongoing clinical studies where they're testing the efficacy of these antibodies in disease prevention. These antibodies contain unusually high levels of somatic hypermutation and other unique features which make it difficult to elicit a consistent broadly neutralizing antibody response. However, there is a clinical need, there's a clinical need to develop a method that could bypass the need to actually elicit these broadly neutralizing antibodies through immunization, which here this paper aims to generate B cells engineered to express broadly neutralizing antibodies, which can then be used to protect against HIV in vivo. So they use CRISPR-Cas9 RNAs to actually initially ablate the endogenous heavy and light chain of B cells. And this strategy is put into place in order to prevent random combinations of chimeric and endogenous B cell receptors, which would result in non-productive B cell production or production of non-engineered B cells. And so they first designed CRISPR RNAs that would ablate the endogenous kappa and heavy chain genes. The CRISPR RNAs, which were designed to ablate the heavy chain gene intron, were positioned strategically to simultaneously disrupt the endogenous heavy chain gene and to optimally position the inserted antibody. So they were ablating the light and the heavy chain, and into that cut site, they were inserting the new engineered heavy and light chain of the new antibody. And their workflow is shown here. So they activate B cells in culture 24 hours prior to transfection, and then post-transfection, they analyze them 24, 48, and 72 hours after. They use a couple of strategies in order to look at whether they've successfully ablated heavy and light chain genes, and that's flow cytometry shown here, as well as TIDE, or tracking of indels by decomposition. And this is a PCR and Sanger sequencing-based method, which is able to identify insertions and deletions and basically quantify these, which were induced by gene editing. So you can see that when they add the heavy and light chain CRISPR RNAs in comparison to control, they're able to successfully reduce the number of kappa or light chain expressing B cells. So this is reduced quite significantly at days 24 and 48, and the expression starts to return at 24 hours and 48 hours, and the expression again is increased at 72 hours. So 24 and 48 are their optimal time points. Further, they show that by tide analysis and flow cytometry, they get very similar results, and they're able to ablate about 70 to 80% of their light kappa chain genes. And when looking at the heavy chain, they're able to ablate 77% of the heavy chain genes. Further, they also had to design a homology-directed repair template, which would be inserted into the heavy chain and would express the newly engineered heavy and light chain of the new antibody. So designed CRISPR RNAs were specifically, again, designed in order to simultaneously disrupt the heavy chain and enhance transgene expression. The homology-directed repair template is a single-stranded DNA and was designed to allow expression of membrane as well as secreted antibodies that would be able to undergo isotype switching and maturation. So the workflow here was again to activate B cells 24 hours prior to transfection, and then 48 hours later, they were analyzed. So here, they're able to show whether cells have been successfully edited by showing by flow cytometry that they are able to bind to their cognate antigen. And when co-transfected with the single-strand DNA and CRISPR RNAs, the cells, they achieved about a 0.1 to 0.4% of cells to actually express the engineered antibody. Because this was quite low, they modified their workflow to include an additional 3 days of feeder culture for the 24 hours post transfection to hopefully expand these engineered B cells. And even though the expression of transgenic antibody was still detected in only about 0.15% of B cells, there was a 6-fold expansion or increase in yield after the 3-day feeder culture. They were also interested in seeing whether they could adopt this system to be used for primary human B cells. And for this, they designed new analogous CRISPR RNAs that would be able to disrupt human light and heavy gene loci. And they used a similar strategy. The single-stranded DNA homology-directed repair template would be replaced by a human and inserted into the heavy chain. And this template, as before, contains homology arms, splice acceptor, and promoter sites. The workflow here was similar to previous workflows. Cells were activated 24 hours post-transfection. They were transfected and analyzed 2 to 3 days after. So here they first aimed to show that their CRISPR RNAs were specific. So when you transfect cells with the light chain CRISPR RNA, you're able to ablate expression, but the heavy chain does not affect the light chain. They were able to show that using their CRISPR RNA targeting the light chain, they were able to ablate B cell receptor expression in 85% of kappa-bearing cells, and also not shown here, but they were able to achieve 64% of cells to reduce the heavy chain expression. Further, they were able to show that between 3 3 to 4% of B cells in human were actually able to express anti-HIV antibodies and bind to their antigen, which is much higher than in the mouse. Here they're showing 2 different antibodies. So the top row is a chimeric antibody, which has limited neutralizing activity. And in the bottom row, there's a potent anti-HIV entirely human broadly neutralizing antibody. And they were able to achieve expression of this antibody on 4% of cells. They also noted that the viability of human B cells was higher than the mouse after transfection, ranging from 60 to 85%. So they went further and they were interested in showing that the edited mouse B cells are able to actually produce broadly neutralizing antibodies in vitro. So what they did here is, as before, they activated primary mouse B cells, they transfected them, and then they IV transferred these into congenitally marked mice. So these mice were then immunized within 18 hours of the cell transfer, and serum was collected on day 7, day 14, and day 21. They then were able to detect broadly neutralizing antibodies in the serum on day 7, shown here in the black line. These antibodies peaked on day 14 and were declining by day 21, which is consistent to a response to an immunization. Interestingly, they also showed that the engineered locus allowed for class switching, which is shown here by the detection of the specific antibody in a secondary IgG1 isotype. So from here, when they looked at IgG serum levels at day 14, they note that actually shorter in vitro culture without feeders provided a higher concentration of antibody in the serum, shown here by the pink and black dots. The broadly neutralizing antibodies were isolated from the serum at day 14, and they were actually tested for their neutralizing activity using 2 different strains of HIV virus. And they noted that 2 out of the 3 mice demonstrated a half maximal inhibitory concentration, or IC50, which corresponds to about 1 in 100 to 1 in 500 serum dilution. And the authors note that this IC50 would be within the range that would confer broadly neutralizing antibody protection, which has been shown in macaques. To summarize the key findings of this paper, the authors demonstrated a CRISPR-Cas9 system that can simultaneously disrupt endogenous heavy and light chain expression and insert an engineered antibody with desired specificity. They were able to transfer the system into using it in human primary B cells by replacing mouse with human elements, where the transfection of human B cells actually had higher efficiency. Also, engineered mouse B cells could be transferred into mouse recipients, and serum antibody titers at day 14 post-immunization had significant neutralization activity against HIV. And to collectively summarize the 2 papers, Wynn et al. Developed a culture system which allows for testing of novel factors involved in bone marrow microniche, and can potentially be used to identify all required factors from transition from short-lived blood antibody secreting cells to long-lived plasma cells. Hartwager et al. Developed a system to produce edited human B cells and demonstrated that broadly neutralizing antibodies, which can be secreted by engineered cells, are capable of producing serum antibody titers associated with protective and neutralizing capabilities. Altogether, this work deepens our understanding of antibody secreting cell biology, which can be utilized to more effectively culture these cells in vitro, and how B cells can be manipulated for therapeutic use. I would like to thank everybody for their time for listening to our journal club, and if you have any questions, we are happy to answer them. Thank you.